Good evening. So now we'll start a new chapter, that is cell biology. So previously called cytology, study the cell. After finding the many refined microscopes, and you have to observe all the plant parts or animal parts, their functions, etc. So now it is called the cell biology. Now first let us see the cell. A cell is a structural and functional unit of all living organisms. Cell biology, a branch of science which deals with the studies on cells, their physiological properties, structure and organelles, that is called cell biology. Now cells are minute, microscopic living entities, invisible to naked eye, could be studied till the invention of microscope in the middle of 17th century. So before that you cannot understand. Even you can understand, you can see the cell and you can see the central nucleus only, no details until electron microscope come into power. So next, between 1632 to 1723, Anton von Leeuwenhoek reported for the first time the structure of bacteria, protozoa and spermatozoa, red blood corpuscles under the simple and compound microscopes designed by him only. That is up to the 500 magnification. The magnification you can see in the microscope. In the eyepiece you can see some 10 or 20 or sometimes 15, and in the objective you have 40, 4500. So based on this you can see the magnification. For example, if the eyepiece is 20, objective is 40, so you have to divide 20 into 40 is the magnification. Like that, and up to 500 power he observed these organs like this spermatozoa, RBC, bacteria and protozoa. So for that, the first time he invented the microscope, he designed the microscope, he is called the father of microscopy. The next, Robert Hooke, a British scientist, observed empty cells in the sections of bottle cork, since they look like the honeycomb like structures which resemble small rooms. He called them as cells. So he is the person who coined the term cell, Robert Hooke. Next, in 1838, Matthias Schleiden, a German botanist, proposed that all plants were made up of cells. In 1839, Theodore Swan, a British zoologist, reported that cells of animals had a thin outer layer, that is plasma membrane, whereas the plants have above the plasma membrane there is a cell wall. So that is the invention of this Schleiden. Later, Schleiden and Swan, between 38 and 39, proposed cell theory. According to this, the bodies of animals and plants were composed of cells and products of science, products of cells. That is the cell theory proposed by them in 38 and 39. Next we have 1858, Rodolphe Virchow. He modified the above theory because of his observations that cells divided and arise from pre-existing cells. So from pre-existing cells, by the division, new cells are developed. In 31, Robert Brown discovered nucleus in orchid roots. In 40, Perkins coined the term protoplasm. This is a gradual discovery of the different items in cytology. What are the aspects of cell theory? So among this we have only important aspects only. It may be very big. And now very important things are, all organisms are composed of cells and products of cells. Cells are the basic and fundamental units of living organisms capable of independent living. all cells arising from pre-existing cells. Each cell is made up of protoplasm with a nucleus bounded by plasma membrane with or without cell wall. So with, that will get in plants, without animals, without cell wall animals. So he made that there is a nucleus and surrounded by the protoplasm, etc., and outer plasma membrane. On outer this plasma membrane there may be cell or cell wall or no cell wall. And all cells are basically similar in their structure and metabolic activities. Function of an organism is the sum total of the activities and interaction of its constituent cells. So inside the cell there are many constituents. The total summation of the function only, it gives the function of an organism. Next, there are exceptions to this theory. So it is given the first one. There is a cell wall. Inside the cell wall there is a plasma membrane. Inside you have a nucleus and also the nucleus and cytoplasm, etc. So that is the theory, cell theory. So what are the exceptions? So now if you happen to see the virus, where what happened? They are between living and non-living and they lack protoplasm. So these are the exceptions to the cell theory. Another exception is bacteria and cyanobacteria. Cyanobacteria, nothing but blue-green algae, they lack a distinct nucleus, no nuclear membrane, no nuclear membrane. And xenocytic fungi like Rhizopus, undivided mass of protoplasm with many nuclei inside. So this is a septate multinucleate condition that is called xenocytic condition that you can find in Rhizopus and Mucor. And red blood corpuscles and also these sieve tube cells lack nucleus. So we are calling as a living cell, but they do not have nuclei. In this RBC, and as well as the sieve tube cells and sieve cells, no nucleus, enucleated cells. Next, subsequent findings added a few more attributes to the cell theory. These are called the cell doctrine or principle. There are structure and function of cells are controlled by DNA and genetic material. Genetic material is the maximum. It gives the transfer of characters and change of characters from one individual to another individual depend upon the nucleotides which are found in the DNA. So this is another. The next, a cell heredity information is passed from one generation to another generation during the cell division. During the cell division, one cell becomes two cells. Either it may be meiosis or mitosis. That transfer takes place through the DNA and also the chromosomes. Sometimes dead cells may remain functional. For example, the tracheids and vessels, they are dead, but they function in a transportation, the translocation of food materials and a translocation of this water, etc. And the horny cells, the animals used to have horn, and those cells are also dead but functional. So these are some exceptions. The next, in 20th century, more detailed information on cells, organelles and their functions were brought about by several biologists with the advent of modern optical instruments and biochemical and molecular techniques. So all living cells on the earth are divided into major groups, namely prokaryotes, for example the bacteria, cyanobacteria, and eukaryotes, all developed from fungi and algae, bryophyta, pteridophyta, gymnosperms, etc. So karyo means nucleus, protos means first. So here no Y, another Y. So prokaryote means the primitive type of nucleus is found there. There no nuclear membrane and no double membrane structures, etc. Those are called prokaryotes. When you come into the eukaryote, they have definite nuclear membrane and double membrane structures are present in the plants. In addition, we will deal some other characters between prokaryotes and eukaryotes. Here you see a prokaryotic organism where you can find at the center you can find this is a nucleoid, not a nucleus, because there is no nuclear membrane. And cytoplasm you can find, and their ribosomes are fine, but these ribosomes are of 70S type, that I will tell you later, 70S type. And mesosome, this is very important, the infoldings of the plasma membrane, that is called a mesosome. You remember, we will have a notes later. So this is the characteristics of a prokaryotic cell. Next we have the eukaryotic cell. Next, eukaryotic cell. So in a eukaryotic cell, automatically you have a definite nuclear membrane that is made up of, here you can find the endoplasmic reticulum, and there is a large vacuole is there, and plastid chloroplast is there, mitochondria there, and Golgi bodies are there. So this is a typical structure of a eukaryotic cell. Now let us see the differences between the prokaryotes and lattice. Prokaryotic cell represent by bacteria, blue-green algae, mycoplasma, and pleuropneumonio-like organisms, etc. Mycoplasmas are smallest cells ranging from 0.3 microns to millimicrons length, while bacteria are 3 to 5 microns. They lack membrane-bound nuclei and other organelles like endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. Ribosomes and centrioles are non-membrane bound organelles found in prokaryotes. And organization is fundamentally similar in spite of wide variety of shape and function only. The organisms, shapes, and different functions. Cell wall is surrounded by cell membrane. No well-defined nucleus is there. So genetical material is DNA, naked, not enveloped into nuclear membrane. Most of the genetic information in single or circular or compact DNA, circular DNA, DNA molecule in the center, incipient nucleus or nucleoid. Since there is no nuclear membrane, the nucleus is called nucleoid. Unique inclusions like mesosomes, I told you already, these are the infoldings of plasma membrane, specialized, differentiated from the cell membrane. Those are the infoldings or invaginations. A special membrane structure, mesosome, by extension of plasma membrane into the cell in the form of vesicle, tubules, and lamellae. Help in the cell wall formation, the uses of mesosomes. Help in the cell wall formation, DNA replication and distribution among the daughter cells during the cell division, respiration, secretion process, increase in the surface area of the plasma membrane, and enzymic contents regarding the photosynthesis, etc. are found in these mesosomes. So in prokaryotes, mesosomes having many functions. Next, chemically complex envelope cell wall consists of tightly bound by 3 layers. Outermost glycocalyx followed by cell wall and plasma membrane. Cell wall composed of peptidoglycan, that is a chemical substance of prokaryotes, peptidoglycan. Next, plasma membrane, this is semi-permeable, similar to eukaryotes, and there interacts outside, interaction between outside. Some bacteria have small circular DNA called the plasmid, that is also very important, in cytoplasm outside the genome. So extra chromosomal DNA, that is called the plasmid, and contribute in the resistance to the antibiotics. It is used in monitoring bacterial transformation with foreign DNA. So this DNA is very important. Next, motile or non-motile these organisms. The motile with the flagellum with the 3 parts, a filament, hook, and a basal body, in addition to pili and fimbriae. The flagella are longer, help for more locomotion, and somewhat smaller are pili and fimbriae. Pili, elongated tubular structures made up of proteins, pillin. The next, ribosomes, non-membrane bonds associated with the plasma membrane, about 15 by 20 nm in size, made up of 2 units, that is 50 and 30, which represent together 70 in prokaryotic ribosomes. These are the site of protein synthesis. The polysomes or polyribosomes, by attaching mRNA, translating mRNA into protein, a bunch of these ribosomes unite together in a chain that is called polysome. So here the diagram is given regarding this prokaryotic ribosome. It is made up of 2 units, and a big unit and a smaller unit. The smaller unit here you can find is the smaller unit. It is made up of 30s and the big one is 50s and the total 70s. So individually 50 and 30, when they compress, that becomes 70. And here you can find a chain of ribosomes attached with a mRNA. This is called polysome. Polysome or polyribosome. Inclusions: reserve materials stored in the cytoplasm membrane, and phosphate granules, cyanophysin granules, and glycogen granules, gas vacuoles in blue, green, purple, and green, and photosynthetic bacteria. These are just stored in this. These are the inclusions of the cell, or reserve materials found in the cell. Now let us see the eukaryotes. Eukaryotes, the new nucleus or developed nucleus or advanced nucleus. They include all members of the fungi, plant kingdom, and protozoans. This protoplast surrounded by cell membrane like protocards, and unlike prokaryotes, most of them contain internal membrane bound organelles, that is double membrane bound organelles like chloroplast, endoplasmic reticulum, mitochondria, Golgi bodies, etc. Each cell organelle plays a unique role in growth and metabolism and contain a set of enzyme catalyzed reactions. So here the prokaryotic, eukaryotic differences are given here. You can see those differences. The first one regarding the size, they are very small, and some are larger, that is 50 microns, and in regarding the eukaryote, most are larger 10 to 100 and some are larger up to 1 millimeter. And general characters, all are microbes, unicellular or colonial, nucleoid, not membrane bound. Here the membrane bound, nucleus is membrane bound. Regarding the cell division, in prokaryotes you can find only the binary fission, whereas there you can find mitosis and meiosis. Flagella in prokaryotes have a simple single rod-like structure, whereas here you can find 9+3 membranes, microfibrils you can find in eukaryotes. Regarding the cell wall, here the cell wall is made up of mucopeptide, I told you already, peptidoglycan, whereas here cellulose and hemicellulose and chitin in fungi are found in eukaryotes. Regarding the organelles, I told you already, the cell organelles here are absent here, whereas here the cell double membrane structures are present in eukaryotes. Regarding the ribosomes, here you can find ribosomes are of 70S type, that is for 50+30, whereas here 80S type you can find. And the last one regarding the DNA, DNA is the genetic material and not found in well organized chromosomes, and whereas here well organized chromosomes are found in eukaryotes. Regarding the cell structure, now let us see, you take a eukaryotic cell, because nothing to tell regarding the prokaryotes, I told you already the characteristics of prokaryotes, but you have to study the eukaryote in detail. There you can find cell wall. This cell wall is made up of 3 layers, I will tell you later regarding the structure, and the middle plasma membrane, I will explain later, and organelles. Regarding the organelles, some are the projecting cells, just like flagella, cilia, etc. Some are not projecting. Regarding the not projecting, the membrane is not absent and membrane bound. Membrane absent and membrane bound. Regarding the membrane absent, you have to see the ribosomes and centrosomes. These centrosomes are found in animal cells. They are absent in plant cells. The ribosomes are found in all cells. And next you see the membrane bound structures. What are the membrane bound structures present in the cell? So regarding these membrane bound structures, some are single membrane, some are double membrane. You have to remember the single membrane structures. Generally questions are asked on what are single membrane structures, or you may be asked one of the following organelle is not having double membranes, like that. So lysosomes, peroxisomes, vacuoles, these are single membrane structures. Regarding the double membrane, we have nucleus, plastids, mitochondria, endoplasmic reticulum, and Golgi bodies. So regarding these plastids are of three types. Skipper coined the term plastid, and here three types. These are the colorless plastids, generally found in the underground parts. The next one is chromoplast, colored plastids that you can find in flowers and fruits, and chloroplast, that is the green plastids. They undergo the photosynthesis, main role of photosynthesis. Regarding the endoplasmic reticulum, some are called the rough endoplasmic reticulum and some are smooth endoplasmic reticulum. Endoplasmic reticulum is attached with these ribosomes, that is called rough endoplasmic reticulum. When not attached, those are called smooth endoplasmic reticulum. So now you can see the diagrams regarding the plant and animal cells here, because simultaneously we should know the difference of plant and animal cells. Before study the cell organelles in detail, we should know the differences between the plant and animal cells. The first one, the blue colored one, is a plant cell. So the important function you see in a primitive cell, that is what's called immature cell or meristematic cell, the vacuoles are very small and sometimes absent. When the cell becomes older and there is a formation of a central large vacuole and leaving the nucleus aside. So the characteristic of a mature cell is having a large central vacuole. So here you can find, this is the nucleus, nuclear membrane, here you can find, and the chloroplasts are there, and mitochondria you can find, and Golgi bodies you can find, and in addition you can have a number of cell inclusions you can find in the plant cell. Very important thing is having the cell wall around the plasma membrane. It's a very important point. While observing the animal cells, here you can find no cell wall, only plasma membrane you can find, and there is a nucleus here. The vacuoles are small. Generally in animal cells, vacuoles are small or sometimes absent. Mitochondria is present, endoplasmic reticulum, Golgi bodies, etc. are present in the animal cell. So the main thing here you can find centriole and centrosomes are present here, but whereas they are absent in plant cells. Centriole and centrosome are absent in animal cells, they are present in plant cells. In animal cell, the centriole is present above the nucleus. The main function during the cell division they involve, and they form the that's called spindle fibers, asters from the two poles. Whereas in plant cells, no centriole and centrosome. Can you guess from which part, which cell organelle gives the spindle formation? Spindle formation in the plant cells. You think well? I will tell you in the next class. So now let us see the differences between the plant cell and animal cell. The plant cell, outer rigid cell wall is there. In animals, no cell wall, only plasma membrane. And plant cells have distinct different shape because of the rigid cell wall. But whereas in animals, there is no definite shape and they will change. Plant cells contain plastids, most of important of these green plastids, which give photosynthesis, whereas the plastids are absent in animal cells. And vacuoles are very small to fewer and larger. Here vacuoles either absent or very rarely we can find small in number. Centrosome present only in the cells of some lower plants, very rare, but all animal cells having centrosomes. Okay, so because of this you can find the cell division as I told you already. So we will discuss this one later, and you guess and what are the organelles which are responsible for spindle formation in plant cells. So regarding the dictyosomes, here you can find two types. Dictyosomes or Golgi complex is dispersed throughout the cytoplasm. That's a very important point. You remember here This is a net-like structure. Throughout the cytoplasm, Golgi bodies are found in plant cells. So those are called dictyosomes. Diction means net-like. They form as a net-like. But whereas in animals, the Golgi bodies are organized here appear shallow saucer-like structures or narrow bowl-like structures, and they form at side. One side are groups. It consists of interconnecting tubules in distal region. So the main difference is in plants, the Golgi bodies which are distributed throughout the cell, and whereas in animals, the Golgi bodies are segregated at some places. And lysosomes are found only in the eukaryotic plant cells. These lysosomes are absent in animal cells. Animal cells are absent. And plant cells is larger than the animal cell, whereas the animal cells are smaller in size. Mostly starch is the storage material, but in animals, glycogen is the storage material. Glycogen is the storage material. During cytoplasmic division, cell plate forms in center of the cell. In animal, this cell plus furrow appears from the periphery to the center of the cell. So very important, this is a very important aspect. I will tell you. After cell division in mitosis, a cell plate forms in the center. Here daughter cells, nuclei, and from center it proceeds to the peripheral side. Center to the peripheral, this is called centrifugal wall formation in plant cells. Whereas if you happen to see in the animal what happened, a furrow develops in the peripheral side, and two nuclei are there, and gradually the furrow proceeds and form two cells. So that is from periphery to the center. So it is called a centripetal wall formation we can find in animal cells. So you remember. So this is the centripetal wall formation is the character of animal, and centrifugal, that is from center to periphery, this is the characteristic feature of the plant cells. So this is given here. During cytoplasmic division, a cell plate is formed in the center of the cell and proceeds to the peripheral sides. Whereas here, during cytoplasmic division, a furrow appears, a depression, a constriction from the peripheral side, and it proceeds to the center, and then we'll get the two cells. That's all. That is the difference between the cell wall formation. So cell in its parts, let us see some characteristics. We will go detail later. Now first is the cell wall. It gives rigidity, shape, mechanical support. Second point, permeable to water and minerals. Next, the plasma membrane. It is selectively permeable, regulates the movement of material across it, and involves in active transport of ions. Next, nucleus. It controls all the cellular metabolic activities, initiates cell division, responsible for heredity, help in transmission of characters. Number 4, chloroplast. They prepare food material by the photosynthesis process. Next, mitochondria, concerned with aerobic respiration. ATP synthesis takes place by oxidative phosphorylation. Next, so it stores ATP, so this is the energy cell of the cell, energy. Next, endoplasmic reticulum acts as a circulatory system for intracellular transport of various substances, and rough endoplasmic reticulum involves in protein synthesis, and smooth endoplasmic reticulum involves in lipid synthesis. Next, Golgi complex. It involves in cell wall development. Main function is cell secretion. Involves in the synthesis of glycoproteins, glycolipids, etc. This is very important. Often is asking in the question examination. Involves in the synthesis of glycoproteins and glycolipids. Ribosomes involve in protein synthesis. Then lysosome help in the intracellular or extracellular digestion. These are the suicidal bodies. Next, vacuoles. They store metabolic wastes and act as osmoregulatory substances. The chromosomes, the last, this one, they transmit characters from parents to children, and having genes, those genes are arranged in a linear manner. Each gene is nothing but a part of DNA, and they carry genetic material, and they just pass the genetic material from one generation to another generation. That is the function of the chromosomes. These are, in short, I told the functions of the cell organelles, and from tomorrow we'll take in detail the cell organelles and their structures. Thank you.